Oh, let me make you a co-host. How about that?
Well, good morning, everyone, and welcome back. Hope you all had a good weekend. Um, we have just finished Unit 1, so I have a little bit of catch up to do in terms of grading some of the work, but we got our second lab up on soils, and I noticed that I do need to put that, turn that into a lab, actual assignment turn-in into, it's just a page right now, so I'll make sure to do that today. Anybody have any questions or concerns before we get started? All right, well, we'll go ahead and get started on Unit 2, which that means it's about time for midterm. So we will be getting into that very soon. So Unit 2 is really all about the plants themselves, and we're not going to get too deep into the weeds, as they say, in terms of, I don't think that you all need to know the particulars of a lot of the botany and chemistry, so I've tried to boil it down to the most basic stuff that I think is important to know in terms of plant science. So we start off just talking about the structure of the higher plants, which the higher plants, we consider those that we most often work with, the flowering plants. All right, so we look at plants and their features and identifying plants, mostly from the mostly from the external structure of plants. And we don't look as much at the internal stuff, even though we will discuss that and look some into the internal parts of plants. We use mostly the external stuff for identification and classification of plants. That has changed a little bit over the, I guess, technological evolution of DNA discovery, really. Now they look more at the genetics, the DNA in terms of grouping plants together, whereas they used to just do it on the shapes of flowers and seeds. So just kind of the first classes, we talk about the phylums or phyla of plants, and pretty much all of our kind of major economic plants, things that we use in agriculture, are mostly in two different phyla. They're in the coniferophyta, or the gymnosperms, and they are in the angiosperms, which is the magnoliophyta, which are the flowering plants. So gymnosperms, not a whole lot of use in agriculture other than for wood, and to some extent maybe some fibers associated with wood. So mostly we're looking really at the angiosperms, the flowering plants. And we look at that phylum of plants as being divided into two major classes. And those are the monocots, or the monocotyledons, which is really classified just because of the seed leaf, the cotyledon. If you actually take a look at the seed itself, that cotyledon is, in a lot of the plants, a lot of the angiosperms, a big portion of that seed. Monocots have one cotyledon, dicots have two. So that's the very easy way to distinguish the two plants. Let's take a closer look here at the life cycle of a monocot. And a good example of a monocot that we use in agriculture is corn, Zea mays. And so we have the corn seed here, and looking at the parts of the seed, we think of it as an individual corn kernel, and inside of it we have these different parts, which we'll look at in a little bit more detail. But mostly made up of an endosperm, which the endosperm is where there's a lot of the food that is feeding the developing embryo. The embryo itself is a good portion of the seed, and then the coleoptile, or really what is the cotyledon itself, is also a good part of the seed. So think of any life cycle of a seed plant, any of the angiosperms. We plant that seed, and corn is one of those examples where we really have to plant it. The way that we've domesticated the corn plant, it has such a tight sheath husk around the kernels and the seeds that you really have to remove it, have to take the seed off of it and put it in the soil in order to get it to grow. Not very often would you have a volunteer corn plant come up from a discarded corn cob. It could happen, but it's not very frequent that would happen, just because it's so highly evolved with people. But anyway, you put that dry seed into moist soil, the seed begins to imbibe or just take up water from the soil, and then it's going to germinate. And germination is kind of a chemical as well as physical process that is dealing with that taking in of the water, the expansion of all the parts inside of it, and the reactivation of those parts inside of the seed. And really, we kind of look at germination as the emergence of two parts: the radicle, which is the primary root below ground, and the plumule, which is the primary shoot above ground. And so we look at that as the first cotyledon, and why it's called a monocot, for what's shooting up above ground. And they're kind of going in opposite directions. The radicle emerges first, and then the plumule emerges, and we actually see the plant growing above ground. And as they enlarge in kind of both directions, they will kind of develop different plant parts coming off of them. And those first primary plant parts don't play as much of an important role anymore. So the radicle grows down through this coleorhiza, this kind of like a taproot, but it's just this area where there's active cell division taking place, and then we have secondary roots branching off of it. And then you can see the cotyledon itself is kind of really just feeding that embryo until the first true leaves appear and it can photosynthesize on its own. So of course, they can develop very long and strong roots as they continue to grow, and a mature corn plant, they say, can develop roots up to 6 feet long. And they kind of special because it's not only developing these regular underground roots, it also develops these adventitious roots. And adventitious roots are those that just appear from anywhere other than the underground portion of the plant. For adventitious roots in corn, we call them brace roots or prop roots, because their function is to basically form and hold up this kind of skinny plant, tall, skinny plant. So looking at some of this in a little bit more detail. First, the emerging plumule is actually protected by that coleoptile, which is kind of just the guarding part. It kind of looks like what you would think of as that first cotyledon, but it's actually just this sheath. And you can kind of see it inside of the seed protecting that plant. And as the true foliage, first true leaves develop, then that coleoptile eventually will fall off. Doesn't need to feed that embryo anymore because it can make its own sugar through photosynthesis. With corn, when the plant gets to a certain size, and generally that's looked at, and we can kind of gauge that on the number of leaves that it has. then it will trigger the formation of the female and male flowers. And the female flowers are the pistillate flowers, because the pistil is the actual sexual part of the female flower that receives the pollen. And in corn, we call those silks. And later we have the staminate flowers, or the tassels forming at the top of the plant. And corn is also an example of a plant that is monoecious, meaning both sexes are on the same plant, but it has what we would call imperfect flowers, because the flowers don't have both male and female parts on the same flower. They're on different parts. So the male is at the top with the tassels. The female, each of those pistils or silks is connected to an ovule, which then becomes a kernel of corn. So each one of those has to receive a grain of pollen from the tassels of the plant in order to make a full ear of corn. Pretty neat. Corn is a grass. It's in the Poaceae family, the grass family, and it's one of the most economically important plant families because it has all of our grain crops in it. Of course, those grain crops are very important for us in terms of calories, and for feeding people, but also for feeding animals. And corn is kind of one of the most important, I think, because it is grown now around the world and used in a lot of different cultures. It is wind pollinated, just like all the other grass. So it's that efficient. As any of you that have grown corn know, you have to plant it in kind of blocks, where you have lots of rows next to each other, so that wind can blow the pollen grains from the tassels to the pistils, to the silks, in order to make that full ear of corn. So each of those individual ovules has an ovary in it that develops into a fruit. And in the grains, the fruits are called a caryopsis, and that's the true seed. And then those kernels mature and dry, and then they're harvested, and we store them over the winter, and we plant them again when the warm season. And so, considering we were just kind of finishing up talking about climate and weather and soils, and kind of how that all goes together, and determining your growing season, how and when you can plant and grow crops, I think it's important to note that corn and most of our annual angiosperms, they are grown here as annuals, and that growing season is determined by frost, because they are our warm season crops. They are not frost hardy. They are frost sensitive. So as soon as we have the first killing frost, that's it for them. If they haven't finished producing those ears of corn, then they're not going to be able to, because that frost will cause the cells to actually rupture and quickly kill the plant, which is coming up. We've had some light patchy frost around here this past week. Not a killing frost yet though, and I'm waiting as long as I can myself to dig up my sweet potatoes, but I don't want them to get frosted on. So right now for us, it's a very important time to be looking at what the low temperatures are each night, to if you're planning on harvesting any crops that you still have in the ground. All right, so let's look at an example of a dicot in the life cycle of it. So since we started with corn, we'll look at beans here, kind of our counterpart in terms of diet, because if we remember talking back about the nutrition aspect, corn and all of the grains are lacking in one essential amino acid, often lysine, which beans supply, and beans are lacking in methionine, which the grain supplies. So by having those two, we get a full set of amino acids, which allows us to make the proteins that our bodies need to function and grow. So anyway, looking at a bean, same deal. We grow it as an annual. We take those dry seeds, we put them in moist soil whenever the danger of frost has passed, and it starts to take in that water, it swells, and then the seed coat will kind of bust open, and the radical emerges, that primary root. So a little bit different thing here going on with the dicots. And if you look at this seed, the seed itself is mostly the cotyledons. So as the radical grows down, we have kind of what we'd call the bean hook or the hypocotyl emerging first. And that's just because it's got these two seed leaves and they're a little bit heavier. So the stem emerges first and has to get enough strength pretty much to basically push those cotyledons, pull them up out of the soil and push them up above that hypocotyl. So then we're developing, it's got two cotyledons. These are always above ground, and they're feeding that embryo until the first leaves can begin to photosynthesize, and then those will fall off and it continues to grow on its own. Dicot, two cotyledons. So, but we've got the hypocotyl, which is emerging first, and it's below the cotyledons. And then we've got the epicotyl, which is above the cotyledons. And that's just any part of the stem that is above that, and then has the true leaves attached to it. Between the cotyledons themselves, as it's emerging, we have this apical or shoot meristem, and that's kind of the growing point where cells are actively dividing. And the meristem, there's a meristem in the shoot. There's also a meristem in the roots as well. But just think of those as areas where the seeds are actively dividing, but also differentiating. So the cell itself, kind of what we think of as a stem cell, both figuratively and literally in this case, it changes depending on, what it's supposed to be. So say it's going to make leaf tissue, it differentiates to make leaves. It's going to make a flower, it differentiates to make flowers. But the primordial cells, the stem cells themselves, are all there in the meristem. And the apical meristem is the one that is basically at the top of the plant. Once everything is right, if we have favorable conditions, then cotyledons fall off and the plants grow and produce and photosynthesize and grow more leaves and continue to grow upward until they become sexually active. Beans are different than the grains in that they are self-pollinated, and they are perfect flowers. So in a bean flower, you've got both the male and the female sex parts, and they are able to pollinate themselves. And our fruits are going to form just basically as long as we have proper environmental conditions. As long as we have the optimal conditions for fruit and flower production, we will get fruit and flowers. Flowers and fruit, I should say. Interesting thing there about beans is that being self-pollinated, they're actually typically pollinating themselves even before the flowers are fully open. Going through the life cycle, though, as pollination occurs, the seeds then mature and dried within the pod, and then we harvest them, keep them, wait until the danger of frost is past, and we can work the soil, and then we plant them again. So that's just looking at the life cycle of a monocot and a dicot. Now let's look into deeper inside the plant, into the cells. So the cell, of course, is the basic structural unit of all living things, plants included. And we look at cytology, which we're not really going to get too much into, but that's the branch of biology that is actually looking at the cells themselves, what is inside of the cells, and all their different functions within the cell. Cells do vary greatly in size as they are differentiated into different parts of different tissues. In all of the cells, though, we have basically like this juice inside, cell juice, and we'll call that cytoplasm. Cytology studies the things inside of cells, including that cytoplasm. That cytoplasm, that juice inside the cell, is basically just allowing the components of the cell to kind of float around into these kind of different compartments within the cell. They have each have their place, and it allows them to move energy through the cell itself. And when we have frost, basically that cytoplasm expands as a result of it freezing, and that causes the cells to just explode, basically. In plants, we have both prokaryotic and eukaryotic cells. And prokaryotic are those that don't have any separate subcellular units, and they're not enclosed in a membrane. We think of those as primitive. So we really only see them in the plant world and things like algae. Most of these prokaryotes are single-celled organisms. Eukaryotic cells, on the other hand, are multi-celled organisms, and they have membranes enclosing them, and each of the organelles inside of the cell, think of them like little miniature organs, have both specialized structures and specialized functions. So looking at some of these inside of the cell, in the plant cell, we've got a few different components than we would have in an animal cell. We got the cell wall here on the outside. It's holding all that cytoplasm in there, and the cytoplasm is kind of flowing within the cell, holding everything in its different places. The few things I just want to kind of point out here in this cell is we have a nucleus in the cells themselves, and we think of that as the brain of the cell. That's where most of the DNA is stored, and that's one of the more important parts of the cell, although I guess all of them are equally important. We'll talk more about these later on, but just to get a look at the cell structure itself. So again, the cytoplasm is kind of this viscous fluid, and it is composed itself of proteins, and it's bound by a membrane that is also composed of these matrix proteins. And we have this kind of movement of the organelles within, and we call that cytoplasmic streaming. So looking at a few of these in a little bit more detail. Again, I don't think you necessarily have to memorize all this, but you probably talked about it in your biology classes before, the different organelles. One of the more important parts is the ER, the endoplasmic reticulum. And that's one of the places where proteins are basically synthesized and created. Proteins are very important because they drive basically all of the plant growth. They are coding for what a cell is going to become. It will have different proteins in it that code for different tissues. Plants are unique in that they have chlorophyll in them, and chlorophyll, within the chlorophyll are these chloroplasts, which are responsible for photosynthesis in the leaves and in some plants the stems, which is basically driving all life on Earth. And that conversion of basically sunlight, water, and some nutrients into, and carbon dioxide as well, into sugar. And so chlorophyll is very important, distinguishes plants from, all other living things. Mitochondria, which they call the kind of powerhouses of the cells, because they are the sites of respiration, and they are taking those proteins and somewhat involved in the synthesis of the proteins, but really involved in the production of the energy-rich compounds like ATP, which is responsible for really the growth of more cells and the respiration of the cells, leading to the growth of the plant and other parts of the plant. We mentioned the nucleus is our kind of brain of the cell, and that's where basically all of the, or most of the DNA is stored within chromosomes. So DNA and deoxyribonucleic acid is kind of our directions for how, the genetic code for how everything is transcribed from DNA and then translated into proteins. So it's really the directions for life, really. We also have a little bit of DNA found in the mitochondria, the cytoplasm itself, and also some in the chloroplasts. So those are also important in passing along those genes and traits. The nucleus is the most important, but these other parts of the cell have a little bit of DNA in them too. The vacuoles, which are a large part of the volume of a cell, is basically just storing water, which we talked about how water is, in a lot of plants, 99% of the tissue of plants is water itself. So it makes sense why the vacuoles that are, storing this water would be one of the largest parts of the cell. We also have some pigments in the vacuoles as well as in the chloroplasts of the cells, and those can impart different colors in the plants. You see blue, red, or purple often as coming from this anthocyanin pigmentation, and the science is still somewhat young in it. I think they've known about this for a long time, but they're finding more and more that the pigments themselves actually also play a role in our nutrition. So we get different things from the different colors of plants too, which is, I think it's kind of neat. So of course we have a cell wall that protects the cells, and it's kind of like a square or rectangular shape in plants. And the cell wall itself is made of this component cellulose, as well as some pectin and lignin. And it's important because it's giving the plant structure itself. It's supporting the plant. We have to have this cellulose in order to have the plant be able to stand up. Between the cell walls are different layers and different membranes, but I don't think for our purposes it's all that important to really look at them in detail. But it is important to recognize some of these chemicals that compose these cell walls and membranes. So in different plants and different tissues, we also get things like lignins, suberins, and cutins. And these are all important for creating those membranes, creating the structure of the cells, and in the case of woody plants, creating the secondary xylem, which really differentiates them from the herbaceous plants. When you cut a plant, they utilize suberins and cutin basically to start healing the wound almost immediately. It's kind of like a plant's skin in a way. Whenever we have large areas of organized cells that have a similar structure, they combine to make the organs of a plant through developing large swaths of plant tissues. And it's really the same as it is in humans and other animals. It's just made up of different stuff. So in all plants, both young and mature, we look at two different kinds of tissue, which we already mentioned. First, we have the meristem itself, or meristematic tissue, which is the actively dividing cells that are then developing and differentiating into other tissues and organs. And then we also have just the permanent tissue, which is what is developing from those meristems. It has differentiated into the different parts, and it includes an epidermis, kind of its exterior skin, and then under that it has three different types of cellular structure: parenchyma, cell, sclerenchyma, and collenchyma. And these become important in terms of propagation of plants. I mean, they're important just for the growth of the tissue itself, but we don't necessarily have to memorize those again, at least not now. So those are simple tissues, and then we have complex tissues, which we'll kind of boil those down into the basic, the xylem and phloem. And we'll talk about those in more detail later, but for now, just think of xylem as transporting mostly water and phloem as transporting mostly food. And they're tubes that basically go all the way from the roots to the leaves, and are connecting cells throughout. In the shoot, we've got this apical meristem, the growing tip of the plant, which is developing into new branches and new buds, which then grow new plants. And this apical meristem is where the plant stores a lot of its hormones, which are basically signaling different tissues to differentiate into different things, telling the cells what to do, whether they're supposed to produce roots or produce shoots. At the other end, we've got the root meristems, which are going to be really at the terminus of each root, the growing points of the root system. So if we look at this image, it just looks at how we have some plants that have a very dominant taproot, and almost all plants do begin with a dominant taproot as kind of that radical. But some plants are more taprooted in that they just develop that as their main root and have a few roots growing off of it. Other plants can be differentiated in that they have more of a fibrous root system, where the roots really are branching in many directions from the very beginning. And that's not really a distinguishing feature of any particular type of plant per se, but in general, monocots tend to have more of a fibrous root system. So our grasses tend to have more like that, whereas the dicots tend to have more of a taprooted system, at least at the beginning. Now I'm looking a little bit deeper into the tissues themselves, into the meristems. We also have a lateral meristem, which produces the secondary growth. And again, we see this mostly, we see it in all plants. This lateral meristem is going to be basically producing the girth of the stem itself. So the stem is growing outward. We got the apical meristem and the root meristem allowing it to grow up and down respectively, with the lateral meristems allowing it to grow in thickness. And these lateral meristems are in this cambium, this vascular cambium inside of plants, which is responsible for forming new xylem and phloem. So again, we can see this in kind of the rings of a plant. If we take a cross section of a stem, then we see these different rings. And one of those is going to be phloem, transporting the food. And then one is going to be the xylem, transporting the water. And in between those is that vascular cambium. We have another layer of cambium in the plants that produce bark, and that's the cork cambium, which just makes that very specialized organ in both herbaceous to a certain extent, but chiefly in the woody plants. Speaking of woody plants, here is a cross-section of a woody plant and where we can really see those growth rings and really get to see that lateral meristem, the activity of it, because you can see it in the growth rings. And we'll see an alternating band of xylem and phloem, which produces wood. And that alternating band is one growth ring, one annual growth ring for a woody plant. So in this case, looking at like a pine, this xylem and phloem is being produced, new xylem and phloem each year. And wood itself is really just dead xylem and phloem. Really the living part of it is just right there in the cambium itself, what is producing the new xylem and phloem, as well as the bark and cork on the outside. So looking more at the simple tissues, excuse me. The epidermis, just like in people, is the outside layer, the skin that it's protected beneath it. And in plants, this is mostly made of this waxy substance that we call cutin. And cutin is in, actually we have the same, you know, substance in us as well, in our skin. It's just a little bit different chemical, I guess, formula of that cutin. But for plants, it's very important because it holds that water that the plants mostly are inside of the plant. So when we look at leaves, and this is just an image of the underside of a leaf right here, we can see that we have these pores on the underside of the leaves that kind of extend through that cutin and open up to absorb carbon dioxide, take in the gases, and to release some of that water vapor. So these are stomata, or stomates, whatever you want to call them. Individually, I guess they would be stomates. Collectively, they're stomata. And these stomata are extremely important, not just for that collection and release of the different gases like carbon dioxide, oxygen, and water vapor, but they're also just important for cooling the plant off. That transpiration, that process of letting that water vapor out through these stomata is what cools the surface of the leaves down. Kind of like us sweating. So the leaf epidermis itself is colorless except for the guard cells and the stomata, which actually contain some chlorophyll in that epidermis, which makes them green. The leaves, of course, are green, but it's not the cutin in the epidermis that has that chlorophyll. It's the cells beneath them. We also have some unique structures that occur on the epidermal cells, specialized in different plants, and these are not in all plants, but in many plants they have different functions, depending on what the plant really has adapted to in its native climate. So here's just a look at some different leaf surfaces here, or different parts of the leaf surface. So in these, we would call these each trichomes, where these epidermal cells are kind of elongated into like a hair-like structure. And so some of these can actively absorb water. Some of these can be used to develop into root hairs on a leaf. Some of these contain chemical compounds in them. So those are the trichomes. Looking at the permanent tissues, first the complex ones, xylem. is very structurally complex tissue that is used to conduct water, and along with that water, the dissolved minerals from the roots and move it to all parts of the plant. So xylem is a very, extremely important tissue for, it does move nutrients as well, but it's nutrients that are dissolved in the water. So it's taking food in a way, but it's taking what food is in the soil. So it's taking those nutrients and minerals up through water and transporting them up into other parts of the plant, all the way to the leaves. And xylem chiefly moves up. So it can move some stuff down, but it's mostly moving that good stuff up to the leaves, where we have photosynthesis occurring. Phloem, on the other hand, is food, but in this case, it's food that are serving as the metabolites that are created through photosynthesis in the leaves, and it's moving it both upward and downward through the stem to anything that needs those sugars, which is really all of the other parts of the plant. The stem itself, the flowers, the roots, all the storage organs. So both the xylem and phloem, they're very complex tissues comprised of all these different components together to form into these complex tubes that basically go the entire length of the plant, connecting it all. So if we look at a cross section of a root here, then we will actually see the xylem and phloem pictured in it. So here's some phloem right here. And this is going to look different in all plants, how they are arranged. Monocots have them kind of more scattered throughout the stem of the plants, whereas dicots, they're more arranged in rings like we see in this image. But nonetheless, we see these bundles of kind of the xylem cells going from the root tips all the way up into the leaves, as well as the phloem itself. And then we have our epidermis here, even on the roots, to protect the roots themselves, and the root hairs being what is really taking that into the phloem, the xylem really, but it has to take it through all of these membranes and layers, and there are very complex processes that are used to take each nutrient in from the root hair through basically the cutin itself and the epidermis, and into the areas where it can transport them up and down. At the end of root tips, we also have a root cap, and right behind that root cap is where we have the meristematic portion. So we'll try to end here talking about the roots. But when we mentioned kind of the soil solution, when we were talking about soil, that's water moving through the soil, but it's also taking in minerals and nutrients, taking in these positively charged ions, the cations, and moving them from that soil solution into the roots of plants. So as I mentioned, it has to go through basically all these different membranes, through these really complex, transport proteins, and it's different for every nutrient. But they're going to basically transfer from the root hair through the epidermis and through the cortex and into, not necessarily into the phloem itself, but kind of in between the phloem and these different membranes into eventually the xylem. So that's how water and nutrients get from soil into the plant. We mentioned the adventitious roots earlier, being those that basically just form from any part of the plant other than the underground parts of the plant. And the adventitious roots have different functions in different plants, whether it is to hold the plant up or whether it's just to actually absorb some moisture or nutrients from the air in some cases. And on that note, we'll end at roots, and then we'll come back on Wednesday and talk about the other structural components of plants. Anybody have any questions before we sign off for the day? I think that was probably mostly some review for you guys, right? I mean, you've heard all this at some point or another. And I think usually what you learn in your biology classes is the most important stuff. So we're not going to hammer on that too hard, but definitely just know the primary components of the plant that makes the structure of the plant and how it gets nutrients throughout the plant, both coming from the roots and going to the leaves, and coming from the leaves and going to the roots and all other parts of the plant. All right, I'll be getting our quiz up for this unit as well and be working on grading our past chapters this week. Be sure to take a look at that lab to get it started. I'll put up the assignment part for it today, so you'll have a deadline and can see when that comes up. Other than that, everybody have a great day. We'll see you Wednesday.
Okay.